We are told the DNA era began in 1953 with a famous photograph of a double helix. A young James Watson and Francis Crick captured the world's imagination by unveiling the structure that allegedly unzipped the secret of life and launched modern biology. But the actual proof that DNA is the chemical carrier of our genetic blueprint was published 9 years earlier, in the middle of World War II. It was a discovery that should have triggered an immediate and total revolution in our understanding of life. Instead, the scientific establishment spent the next decade in a state of collective denial. While the double helix is now a global icon, the man who first identified its function, Oswald Avery, remains a figure of quiet tragedy in the history of science. Working out of the Rockefeller Institute for Medical Research in New York, Avery was not a brash young climber. He was a 60-something physician scientist, widely known for his legendary caution, his meticulous white lab coat, and a total refusal to speculate. In February 1944, Avery and his team, Colin MacLeod and Macklin McCarty, published their findings in the Journal of Experimental Medicine.
The paper was titled with typical modesty, but its technical implications were world-shattering for anyone paying close enough attention. They had solved a mystery that had puzzled biologists since 1928, the nature of the transforming principle. They proved that this substance, capable of permanently changing an organism's traits, was deoxyribonucleic acid, DNA, and not the proteins everyone expected. From a purely biochemical perspective, Avery's 1944 experiment was flawless. It was far more rigorous and precise than the isotope experiments that would later win the Nobel Prize for others in the 1950s, using a strategy of total biochemical elimination. Yet, for nearly a decade, his work was met with a wall of institutional skepticism. The top biochemists of the era simply could not accept that a molecule as dumb and repetitive as DNA could hold the complex code of life. Avery didn't arrive at his conclusion through a flash of intuition or a pretty model. He spent over 10 grueling years meticulously isolating the biochemical components of the pneumococcus bacteria, the deadly pathogen responsible for pneumonia and countless hospital deaths.
He and his team subjected the bacteria to a brutal interrogation. They used specific enzymes to destroy every possible candidate for the genetic code. First, the proteins, then the RNA, then the fats and the complex sugars that formed the cell wall. In each case, the bacteria could still pass on their traits. Only when they used an enzyme called DNase, which specifically destroys DNA, did the transforming principle stop working. The proof was absolute. The code was located in the DNA. For Avery, the results were clear. DNA was the gene. But he was up against a powerful scientific dogma that favored complex proteins over simple acids. He had the empirical truth, but he lacked a narrative that his peers could stomach. He eventually died in 1955, never having received the Nobel Prize he clearly earned. His legacy was permanently overshadowed by the younger, more charismatic scientists who stood on his broad biochemical shoulders to build the modern world. This wasn't just a mistake of history. It was a fundamental breakdown in how science processes evidence. To understand why Avery was rejected, we have to understand the protein paradigm that dominated the early 20th century. In the 1940s, the smartest people in the world were convinced that the genetic code was written in a language far more complex than anything DNA could offer. That impossible theory was driven by what historians now call the protein paradigm. In the 1930s and 40s, biochemists were entirely convinced that proteins had to be the carriers of genetic information, simply because they were the only molecules complex enough for the job. Proteins are built from 20 different amino acids, which means they can fold into a nearly infinite variety of intricate shapes and structures. To the brilliant minds of the era, this vast chemical vocabulary was exactly what a cell needed to store the complex instructions of life. And then there was DNA. At the time, the leading structural model was Phoebus Levine's tetranucleotide hypothesis. It depicted DNA as a painfully simple, repetitive molecule, just 4 bases arranged in a monotonous, repeating square. It was considered nothing more than a boring structural scaffold. If proteins were a highly sophisticated data storage system, DNA was viewed as just the structural glue holding the cell together. The idea that this repetitive string of chemicals could harbor the inherited traits of every living organism contradicted the basic rules of chemistry as they were understood.
But nature has a habit of breaking established rules. The first crack in the protein paradigm didn't come from a chemist studying DNA, but from a British bacteriologist named Frederick Griffith, who was simply trying to understand pneumonia. In 1928, Griffith was experimenting with 2 strains of pneumococcus bacteria. One strain was completely harmless. The other was coated in a smooth sugar capsule and was highly lethal, capable of killing a laboratory mouse in a matter of days. Griffith took the lethal bacteria and destroyed them with heat, effectively turning them into a dead, harmless biological soup. When he injected this dead mixture into mice, they survived, exactly as expected. Dead bacteria cannot cause an infection. But then he tried something strange. He mixed the dead lethal soup with the living harmless bacteria and injected that combination. The mice died. And when Griffith examined their blood, he found it swarming with living, fully lethal bacteria. The harmless strain had somehow learned how to manufacture the deadly sugar capsule. A physical trait had been passed from the dead cells into the living ones, fundamentally rewriting their biology.
Griffith called this mysterious transfer the transforming principle. The result was incredibly bizarre. Griffith did not know what the chemical was, and many of his peers doubted that bacteria even possessed genetics in the same way as complex plants or animals. It was easy to dismiss this as just a strange metabolic reaction to stress. Yet, the data was undeniable. Something physical, some highly stable molecule had survived the heat and carried a precise set of biological instructions across the cellular boundary. The instructions were real, but the messenger was completely invisible. This strange bacterial phenomenon caught the attention of Oswald Avery at the Rockefeller Institute. Avery was a meticulous researcher, not prone to wild theories, but he recognized that Griffith's transforming principle was not a mere anomaly. It was a biological signal waiting to be decoded. If he could isolate the exact chemical responsible for delivering that lethal message, he would definitively prove what genes were actually made of. It was a problem that required stripping away the bacteria piece by microscopic piece. Avery would spend the next decade doing exactly that, employing a method of biochemical brute force that was so rigorous it would leave the scientific establishment with nowhere to hide. Avery's strategy was not a sudden stroke of genius, but a brutal, years-long process of elimination. If the scientific establishment firmly believed that protein carried the genetic code, he was going to systematically destroy it and force the cell to confess the truth. Working at the Rockefeller Institute alongside Colin MacLeod and Macklin McCarty, Avery focused entirely on an anomalous bacterial trait. They were hunting the transforming principle, a mysterious, invisible agent
that somehow transferred virulence from dead bacterial cells into living, harmless ones. Their approach was elegantly simple in its logic, but agonizingly difficult to execute at the lab bench. Without modern centrifuges or automated sequencers, the team spent the late 1930s and early 40s grinding down immense vats of bacteria to purify the chemical soup. After exhausting physical extractions, they managed to isolate the active substance. It materialized in their vintage glass test tubes as a clear, highly viscous liquid that could be delicately spooled onto a glass rod. To the naked eye, this stringy material was the physical embodiment of heredity.
But the biochemical question remained violently contested. What exactly was this substance? Was it the much-championed, deeply complex protein? Or was it something else entirely? To find out, Avery's team deployed a method of enzymatic interrogation. They introduced specific, highly purified enzymes into the viscous extract, acting like chemical assassins targeting a single molecular profile. First, they added proteases. These were enzymes specifically evolved to ruthlessly shred proteins into inactive fragments. If protein truly held the genetic code, completely destroying it should have stopped the bacterial transformation dead in its tracks. But it did not. Even with the proteins chemically obliterated, the stripped-down substance continued to pass on its lethal genetic instructions perfectly. The team meticulously documented the failure in their Rockefeller archives. Next, they turned their chemical weapons on RNA, deploying RNases to eliminate it from the equation. Once again, the genetic transfer continued completely uninterrupted. They were stripping away the biological suspects, one by one, shrinking the margin for error.
Finally, they introduced a relatively rare enzyme called DNase. Unlike the others, DNase had exactly one specific target. It only broke down deoxyribonucleic acid. The moment DNase was unleashed into the viscous sample, the genetic transformation completely halted. The active agent had been irreparably destroyed. The bacteria could no longer transfer their traits. By methodically eliminating every other chemical candidate, Avery's team had successfully isolated the true blueprint of heredity. It was definitively not the infinitely complex protein that the establishment adored. It was DNA, the precise molecule that the world's leading biochemists had dismissed as a dumb, repetitive structural scaffold. Decades of Avery's career culminated in this flawless empirical proof. Even today, as we observe the 2026 commemorations of World DNA Day, Avery's unglamorous persistence remains the historical gold standard for isolating molecular function. He didn't just guess, he proved it by subtraction. The evidence was successfully isolated in a test tube, peer-reviewed, and printed clearly in the Journal of Experimental Medicine in February 1944.
By all logic, the case should have been definitively closed. But when that landmark paper hit the desks of the scientific elite, the reaction wasn't applause or a sudden parody. That institutional denial was not a sudden explosion, but a quiet, suffocating silence. In February 1944, Oswald Avery, Colin MacLeod, and Macklin McCarty finally published their results in volume 79 of the Journal of Experimental Medicine. The document is a masterpiece of biochemical rigor. It detailed a decade of exhausting, methodical elimination, proving that only when you destroy a cell's DNA do you destroy its ability to pass on inherited traits. The data was bulletproof, yet it failed to trigger an immediate scientific revolution. The reasons for this failure expose the fragile, human machinery of how science actually operates under pressure. The first major barrier was simply geographic and disciplinary. The Journal of Experimental Medicine was the premier venue for clinical disease research. It was not a journal of genetics. The theoretical biologists, the experts mapping out hereditary mutations in fruit flies and plants, simply did not read it. To classical geneticists of the 1940s, bacteria were not considered valid models for heredity. They were viewed merely as pathogens.
It was not even universally accepted that bacteria possessed genes in the same way that complex organisms did. Compounding this disciplinary blind spot was the reality of global conflict. The Second World War had severely fractured international scientific communication. Mail ships carrying academic journals across the Atlantic were frequently delayed, intercepted, or destroyed. The physical distribution of Avery's breakthrough was severely throttled. But the deepest reason for the muted reception did not come from German submarines or disciplinary boundaries. It came from Avery himself and his absolute, uncompromising scientific restraint. Avery possessed an extreme, almost paralyzing modesty. In an era where researchers often rushed to claim grand theories, Avery refused to market his discovery. He believed the raw data should speak entirely for itself, without any theoretical embellishment. If you examine the 1944 paper, you will not find the phrase universal code of life. Instead of boldly declaring that DNA was the gene, Avery intentionally hedged his conclusions. He wrote only that the transforming principle was, quote, a nucleic acid of the deoxyribose type. He provided flawless empirical proof for one specific strain of pneumococcus bacteria, but he explicitly stopped short of claiming a universal biological law. By refusing to overstep his immediate data, he inadvertently gave his critics a massive linguistic loophole. The historical disconnect is staggering. Today, on April 25th, 2026, the scientific world celebrates World DNA Day. The public focus almost entirely gravitates toward the iconic 1953 double helix image as the dawn of modern biology. But recent historical retrospectives and scientific institutions now recognize Avery's 1944 paper as the true gateway to the molecular era. Without Avery proving what the genetic material was, the later rush to discover its physical shape would never have happened. Yet in 1944, his extreme caution, combined with the entrenched belief that proteins were the real genetic actors, gave the scientific establishment the breathing room they desperately needed. They read his flawless data, but they rejected the paradigm shift.
Avery's absolute scientific integrity cost him immediate historical recognition. His modesty gave the skeptics the ammunition they needed to launch a counteroffensive. And the most dangerous attack on his work was not going to come from
The harshest criticism didn't come from a rival university. It came from Alfred Marsky, a brilliant and highly respected biochemist working in the exact same building. Marsky was a protein expert, and he simply refused to accept that a basic molecule like DNA could carry the instructions for life. Marsky did not accuse Avery of fabricating data. Instead, he weaponized the technical limits of 1940s chemistry. He focused his attack on a microscopic sliver of doubt within Avery's own rigorous extraction process, demanding a standard of proof that was practically impossible. Avery's sample was extraordinary for its time, at least 99.9% pure DNA. But in biochemistry, flawless perfection is a myth. Marsky seized on that remaining fraction of a percent, arguing that a trace impurity was hiding inside the test tube. To the scientific establishment, proteins were complex, powerful machines. Marsky argued that the tiny trace of contaminating protein in Avery's extract was actually a hyper-potent genetic agent. In his view, the massive volume of DNA was just a dumb structural scaffold.
holding the real intelligence.
This became known as the Contamination Argument. It was a perfectly rational hypothesis that mutated into an institutional blind spot. Scientists were so deeply committed to the protein paradigm that they invented increasingly absurd chemical epicycles to protect it. They willingly preferred the existence of an invisible, magical protein over the visible, proven DNA that Avery had meticulously isolated. For Mirsky, it wasn't personal malice. It was a rigorous defense of the known laws of chemistry against a claim that seemed chemically impossible. Strictly speaking, Mirsky was right. No biological sample in that era could ever be guaranteed 100% pure. If Avery had possessed modern sequencing tools, he could have rendered the contamination argument instantly void. But Avery only had enzymes. He had already proved that adding DNase, an enzyme that specifically destroys DNA, completely stopped the genetic transformation. If the active agent was a hidden protein, destroying the DNA shouldn't have mattered. Logically, Avery's enzyme tests had already defeated the contamination argument. But logic is rarely enough to overthrow a dominant paradigm.
Mirsky published relentless critiques in scientific journals, aggressively challenging the purity of the samples, until the debate infected the broader community of geneticists. Avery, who was notoriously modest and cautious, found himself exhausted. Instead of being celebrated as the pioneer of a new biological era, he spent his final active years repeatedly defending his flawless data against colleagues who simply refused to concede. This is a textbook example of how a dominant scientific framework defends itself. When presented with anomalous data, the establishment does not immediately change its mind. It weaponizes the margin of error. By demanding an impossible standard of absolute purity, the establishment avoids grappling with the structural threat to its core beliefs. For 8 long years, the contamination argument successfully held the line. The world's top biochemists managed to stall the DNA revolution by pointing at a microscopic sliver of doubt. But science didn't finally accept DNA because Avery got better or cleaner data. They accepted it because they got a better story. In 1952, a much messier experiment involving radioactive isotopes in a that theoretical dam finally broke with an experiment that was actually less precise than Avery's. In 1945, Alfred Hershey and Martha Chase used a standard Waring kitchen blender and radioactive isotopes to track exactly how bacteriophage viruses infect bacterial host cells. They tagged viral proteins with radioactive sulfur, and they tagged viral DNA with radioactive phosphorus. After running the blender to shear the empty viral shells off the bacteria, they found only the radioactive phosphorus inside the infected cells. The virus was injecting DNA. Suddenly, the scientific consensus shifted. The world was finally ready to accept that DNA was the true genetic material. But from a strict biochemical standpoint, this celebrated blender experiment was actually much messier than the meticulous enzyme purifications Oswald Avery had published 8 years earlier. Avery's 1944 sample was exceptionally pure, reducing trace proteins to less than 0.1%. By contrast, Hershey and Chase's viral ghosts still left behind a significant measurable amount of contaminating protein. Yet, it was their radioactive data that triggered the paradigm shift.
Why did a messier experiment succeed where a chemically flawless one failed? It came down to timing, psychology, and technology. By 1952, the post-war scientific community was completely captivated by nuclear science. The radioactive isotope tracking simply felt far more modern than Avery's classical chemistry. Furthermore, the theoretical resistance had naturally begun to erode.
biochemist Erwin Chargaff had recently proved that DNA was not just a dumb, repetitive string of 4 equal bases. The old tetranucleotide hypothesis, the very assumption that blinded Avery's critics for a decade, was finally dead. The scientific community didn't just wake up and discover the truth in a blender. They finally became psychologically ready to let proteins go. They accepted the proof that Avery had already handed them. The following year, in 1953, Watson and Crick published the famous double helix structure. That elegant double helix gave the world a beautiful, comprehensible shape. It became the ultimate visual icon of modern biology, and it secured the Nobel Prize.
But understanding the geometric shape of a molecule is fundamentally different from discovering what the molecule actually does. But the empirical record outlasts the establishment's memory. Today, as we mark World DNA Day on April 25th, 2026, the scientific community is openly reckoning with this institutional blind spot. We are recognizing the 82nd anniversary of the true foundational paradigm shift. Every modern genetic ancestry test, every CRISPR edit, and every mRNA vaccine traces its intellectual lineage directly back to that viscous, clear liquid at the Rockefeller Institute. The knowledge that the helix actually holds the blueprint of life started with